Hello and good afternoon, everyone. Thank you very much for joining us for this OnlyCells webinar on antibody identification. It's great to have so many of you joining us today. Before we begin, just a few housekeeping rules. Your cameras and microphones have been disabled to help keep the session disruption-free. That said, your participation is very much welcome, so feel free to drop any questions in the chat or use the Q&A function as you see fit. Helen has said that she'd like the questions to be at the end, so if you put them in the chat as and when you want, then we can address all your questions right at the end. And please also note that this webinar is being recorded. By attending, you consent to the recording taking place. The session will be uploaded to YouTube after the event for those who would like to catch up or anyone who isn't able to join us live. And so this webinar came about thanks to many of you. I've had lots of messages from people asking for something on antibody identification. So I thought, why not make it happen? And from the start, my objective for this was it to be a learning experience that's useful no matter where you are in your transfusion science journey. So whether you're just at the start or you're brushing up or looking to deepen your understanding, hopefully this will be for you. And I knew there was one person who could really bring that to life. So I reached out to someone who I've known for a while and truly respect, both as a scientist and as an educator, Helen Thom. And yes, I may have been a little bit cheeky. I didn't ask her to run a single webinar. I asked her if she would be willing to do a full 3-part series. So the idea behind this was to build each session on the last so that we could take a deeper and more structured approach to learning about antibody ID. Unfortunately for us, Helen said yes, so here we are today with session number 1. For those of you who don't know Helen, let me give you a quick introduction to her. Helen Thom is a registered biomedical scientist who began her career at NHS Blood and Transplant as a placement student in 2012. She secured a trainee BMS role in the red cell immunohematology lab, specializing in transfusion science. And after about 7 years on the bench, she moved into the patient blood management team, where she worked on education initiatives. Following this, she had a project management role in the hospital customer services department. And now Helen is part of the Transfusion 2024 program, helping to develop a collaborative models between NHSBT and hospital transfusion laboratories. She's also a member of the Institute of Biomedical Sciences Specialist Advisory Panel for the Transfusion Science and is a strong advocate for the role of biomedical scientists. She's passionate, experienced, and brings a really thoughtful approach to teaching. And we're lucky to have her to lead this series. So with that, a huge thank you to Helen for being here, and I'll hand over to her to get us started.
Oh, thanks, Anas. That was a very lovely introduction. Thank you. And thank you for inviting us. I do really enjoy talking about this, so I hope you all find it really useful. We'll just make a start. So today's session, so as Anas mentioned, we are going to be doing a few of these, but today's one, we're just going to cover the basic principles. So just right from the start, and we're going to look at some of the key testing tools. We're going to look at inclusion and exclusion methods so that you can interpret your panel results, and then look at how the phenotyping plays its part as well. We have got some polls in this, so I'd really appreciate it if you can take part. I know not everyone will have access to them, but for those of you who do, it'll be great just to sort of get involved. So why is antibody identification so important? So we're not just talking about ABO and RHD. There are hundreds of blood group antigens that we need to be thinking about. And your red cell antibodies that can be produced either through pregnancy, transfusion, or transplantation. You can obviously have your naturally occurring ones as well. Immediate or delayed transfusion reactions, so we are wanting to avoid these. So the basic principles. So when I was a trainee, sort of 12 years ago now, 12, 13 years ago, I remember my manager at the time saying it's bucket chemistry. It's basically a drop of plasma and a drop of red cells, and that's stuck with us. And that is what we're doing. Doesn't matter what test you're doing, you are just mixing things together and hoping for the best. So what you've either got unknown antibody with a known antigen, and this is what antibody identification, which I'm going to talk about, or we've got a known antibody with unknown antigen, and this is your phenotyping. So you're using these tools to try and find out what it is. And then you've got your antibody ID, your antibody, we know it'll bind with its corresponding antigen. So when we're doing antibody ID, we're just mixing them together and with known and unknown, and it helps with to identify them. One of the tools that we use is the indirect antiglobulin test. So when you're mixing the patients, when you want to know what patients' antibodies are contained within their plasma, we'll use donor red cells to go with this, and these antibodies will bind, they'll sensitize the red cell, but IgG are too small. They can't cause direct agglutination when you're performing your antibody identification. So we just need to give it a bit of a hand, and in order to do this, you use a bridging antibody. I've opted for the time bridge here because you may be able to tell I'm from the northeast of England. Obviously there aren't red cells stuck on the time bridge, but just to give you that understanding of what it is that's happening. So you've got antibodies in the plasma, you've got your donor red cells, you need something to bind them together to cause agglutination that can let you know if the antibody is present. And this is the anti-IgG, or what we would most be familiar with is the anti-human globulin. So this can be a liquid reagent that you would add if you were doing tube IAT, or it's why your cards are green when you're doing your IAT, because that reagent is impregnated into those cards. This can be used for your patient's antibody identification. So it's the patient's plasma versus donor red cells. But when we do this for phenotyping, because you can use IgG for phenotyping as well, it's the patient's red cells with the known anti-sera. So conversely, you have your direct agglutination. So IgM can bridge the gap, so you don't need to add a bridging antibody to this because IgM is huge. I remember this from uni, was IgM massive. That's how you remember what the M is, and that can actually cause agglutination. So you don't need to use anti-human globulin when you're doing this. And this is used for your blood grouping. So when you're doing your ABO and your D, in any sort of IgM anti-sera, and you can use it to look for cold antibodies as well, because it's very good for that. So the first poll, if you can launch the poll, please, Anas, is can you think of some of the tools at our disposal when we're performing antibody ID? So we'll just make a little bit of a word cloud here. Just type in what you think you might want to use. We'll just spend a minute sort of going through that. So I'm going to type in your panel cells, for example. So that would be your tool. So anything you can think of that you use on the bench. Hopefully that'll work. And I'll just give you all a minute to put that in. Hopefully that's working, and that's because I can only see my response on the page. I don't know if any other responses have gone in.
Yeah, so we've had about 12 or so responses at the moment.
Oh, fab.
So lots of people have said cells, screening cells, panel cells, IAT panel, red cells, pipettes, gel technology, donor, and cards at the moment. We'll give it a couple more seconds and then I will stop the poll.
Thanks. Oh, it's a shame I can't see them. I don't know why it's not letting us. It normally does, but
yeah,
it's one of those things. So quite a few responses there. So yeah, so the things that you've mentioned there, Anna, I know these are all things. So thank you for taking the time to put those in. So some of the things I'm going to talk about here, one is patient history. And I have to admit, I've got to give my friend Jill credit for this because I was telling her that I was doing a talk the other day and I'd listed all of the things here and I hadn't actually put the patient history. And she went, oh, patient history. And that is really important. We need to know about the history and everything of our patients to help with the investigation. You've got your antibody screen, you can use grading. So these are all things I'm going to talk about today. So I've got our panels, the cells, you know, pipettes, they are tools, they are literally the tools that we have at our disposal. We couldn't do a job without those. Phenotyping, genotyping, your DAT and Elution. So it's going to be a whistle-stop tour, but we'll try and get through everything today. So the first tool at our disposal is the antibody identification screen. So I'm really visual, so I'm hoping this is helping everyone because I've just thought, how do you, you know, we can stand and talk about it, you could write an essay on it, but how do you, to me, how do you see what is actually happening? So we've got a patient sample there. So we've said, you know, at the beginning, it's about mixing the plasma and the red cells together. So we've got a patient plasma. We want to see if there's any antibodies in there. So we'll mix them with some red cells, and because these are our screening cells, we know the phenotype of those. So that's that unknown plasma versus the known red cells. We're using our card, and in this case, we're using IAT because it's got the green cards in there. We incubate them at 37 for 15 minutes, and then we centrifuge them. But what's happening under here? And don't forget, we've got a bridge there. So if you looked underneath this, you would have your patient plasma with your antibodies, if they're there. You've got your red cells. Then when you're incubating them, the antibody is binding its corresponding antigen. So here you can see, what's happening is when you use a bridging antibody, you're actually getting agglutination. So the first two cells are positive because the antibody's bound its corresponding antigen, whereas there isn't a corresponding antigen on the bottom cell, so that's negative. And then this is what it looks like when you actually are reading your cards, whether you've done it manually or you're doing it using automation. One of the things we'll do is when we'll move on to grading here. So this is really useful because you need to grade your strength of reaction, and this can actually assist you with antibody identification. It'll help you determine what the strength of the reaction is. So the tops, if anyone on the call does tube technique, there's just some different grades of reaction there. And on the bottom is probably what we're most familiar with, ranging from 5 all the way down to a sort of mixed field reaction. We can use these to see if there's multiple antibodies, or you can get enhanced reactions, and it can even show dosage, all of which I'm going to talk about. So if we go back to our screen here, you've got screening cells 1, 2, and 3 there. You've got your screening antigram, and you would record the results there. So for me, I would call these 3+ reactions. So I'm going to write down that my results are 3, 3, and 0 for my negative. You might record it as a dash, but I would put 0. That's just how I do it. And that's how you work out what your screen results are. But that doesn't really tell you what's going on there. It just tells you that there's an antibody. It might not be enough to actually give you the antibody identification. So thankfully, we'll have our panel cells, which is the next step. So you would do your group and screen, go on to your antibody identification. Your panel cell, for those of you who don't know, they're not just random, they're made up in a specific way. They are made from donor red cells. So when you donate blood in the UK, and probably everywhere else as well, they're made from 8 or more group O donor cells. So when you donate blood, it may go directly to a patient, or you might turn into a reagent, which is quite helpful. They're set up in such a way that you can get your commonly encountered antibodies identified using those panels. So it's got at least 2 antigen positive and 2 antigen negative examples of those. It has to have at least 1 R1R1 and 1 RWR1. So these are big Z positive, D positive, little e positive cells, and one has a CW in it as well. Between these two cells, they've actually got to express the K antigen, big K, little k, Duffy A, Duffy B, JK, JKB, big S, and little s. So it isn't just random. These cells are deliberately made in this way. You need to have one example of an R2R2 cell, R-r, and R-r, and you can see the different phenotypes there. You've also got to have 3 D negative cells, which are the little r phenotype. One of those has got to be K-pos, and between them, they have to show homozygous expression of little k, your kid antibodies, your S's, and your Duffys as well. So it isn't just random. You also can have papainized cells on your panel, and I'm going to talk about the impact of papain, which hopefully, if you don't know already, we'll cover that as well. And the reason that we do this is because not only have you got to identify an antibody, but once you've identified an antibody, you need to be able to exclude the presence of any further clinically significant antibodies, because you're wanting to avoid the patient having a transfusion reaction. So we as scientists need to do everything that we can to make sure that we've excluded everything, so then the blood that we're selecting is appropriate for our patient. And you can see there, the red cells, it's saying here, in particular, the presence of anti-JK, JKB, big S, little s, Duffy A and Duffy B should be excluded using red cells that have homozygous expression for the relevant antigen. And when you're doing your exclusions, you only need one example. to exclude upon. So the next poll, please, Anas, is what do we mean by homozygous expression of the relevant antigen? Is this a panel cell that shows double dose expression? Or is it a panel cell that shows single dose expression? Or you aren't sure? And it's okay if you're not sure. Please just pop them in. And all results are anonymous as well. So don't worry if you're getting there.
The poll should be up now.
Yeah, I can see that. Thank you. So far we've got 81% going for double dose expression. A couple of people aren't sure there, and some single. I'll just give it another minute. Single dose. Oh. Right. If we can just close that there. Thanks for everybody for answering. So what it means is the cells actually shows double dose expression. So 78% of you getting the right answer there. If you got the wrong answer, don't worry about it. This is why we're having this session. So you can see hopefully on the panel cell there. So this is what I mean by double dose expression. So this cell here is JKA pos, it's JKB negative, which means the person who on this cell, they have inherited 2 copies of the JKA gene. So there's effectively twice as much JKA antigen on there than a single dose cell. But what is a single dose cell? So the single dose cell, so you can see on the bottom there, these are what we call sort of heterozygous. And what this means is it's got 1 copy of JKA and 1 copy of JKB. So it's only a single dose of JKA. So you can actually get weaker expression. And this is really helpful when you're looking for your antibodies. If you have weaker reactions when you're doing your grading, that you can actually see that you've got homozygous expression. So if you get they were got on the top a 2+ reaction by IAT, and on the bottom line it's actually only picked it up in Enzyme, so that's a useful tool to help you identify antibodies as well. So that's what we're looking at with panels. Like I said, we'll perform IAT on these. We can use non-enzyme treated cells, so that's your normal sort of panel cells, and you can use your papillary cells. You could even do a cold panel if you wanted to as well. So this is our panel sheet. So you can see the bridge there at the top, we use an IAT. And I've just put a note on the side there just to highlight that these are made up in that particular order of your group O donor cells. That, you know, we've got the R1, we've got an R1 with a CW, you know, R2. So next time you look at your panel cells, you'll be thinking, oh, actually, these are made up in that way because they have to be. It isn't just random. They may not necessarily be in this particular order. This is one of the NHSBT panels that I've used, so it might look slightly differently in your laboratory. One of the things they have highlighted is that we have enzyme panels there. So in the reference lab, we tend to do enzyme IAT because that's really good at detecting KID antibodies, but you just may use an enzyme neutral card. So just to be mindful when you are performing your antibody IDs in-house. Now, for me, the best way to think about enzyme is you'll learn that I have a bit of a thing for SpongeBob. I do like SpongeBob. So I'm looking at Patrick here. And if you think of your enzyme panels, if Patrick was to come along, it's going to take some of those antigens away. So you might hear that antigens can be sensitive to papain. So I just got this picture here just to demonstrate how this works. So if you think of your red cell, it's the antigens of having huge trees, or Squidward in this case, who is a KID antigen. And Squidward's trying to get a bit of a suntan, but actually some of the other trees are in the way. So what we do is Patrick comes along and Patrick's going to get rid of those trees because Patrick is papain. So then what happens is came along and those ones that are the antigens that are sensitive to papain, Patrick's knocked them out the way. Now Squidward can get a really good tan. And this is sort of steric hindrance. So it's moving some of the antigens out of the way. So your antibody can actually bind. So this is sometimes why you get those stronger reactions, particularly with your RH and KID, that reactions are enhanced when you use papain. And this is because the M&S and the Duffys are out of the way, so there's more room for those antibodies to bind. With your KEL, I've seen it where you can get increased strength, but not always. Sometimes it is just the same strength. So just things to bear in mind. So when we're going through an antibody example, this is how you would get your exclusions, or this is how I would get my exclusions. So record all my results. You can see I've recorded some results there in the IAT and Enzyme using the grading from the cards that I would have done when I've performed my antibody ID. I take my pen and I start at the end, and what I do is I go down until I've got a positive reaction. So you can see on this cell, sorry. So on line 2, I have a positive reaction. So I go down to that positive reaction. I look to the side, can I exclude this antibody? There is a positive reaction there, so I can't exclude it. So I continue down to the next positive reaction, and then I move along. Oh, great, this time it's negative. So using the BSH guidelines, I would say I've got a homozygous expression of GKB here, and it's negative, so I can actually exclude the panel on this. So then following the guidelines, you only need 1. I personally like 2, but when you've come from a reference lab, you see all sorts, so I think you're just a bit more cautious. But obviously follow your local policies when you're doing these. So I go down to the next one, that's negative, great, I can get rid of anti-GKB. And then I cross it out at the top so that I know it's gone. I've dealt with that column, I can move on to the next. So sort of here's one I did earlier. So I've got rid of GKB, great. And then I'll just quickly go through everything else because we'd spend the whole time just me talking about doing exclusions. So hopefully that's helped you to see how I would perform those exclusions. So now what I'm looking at is I've gone to the end and I can't get rid of D, I can't exclude D. So now I'm going into inclusion territory where I'm thinking, hang on, this is really looking like it's an anti-D. And using me grading and using my knowledge of how antibodies behave, if they're papain sensitive, if they're enhanced by papain, in this case, RH antibodies are. And if you wanted to know what is, you can check your BSH guidelines, or it might be incorporated in your local SOPs. So this is really looking like it's an anti-D, and I know that it's been enhanced in the papainized cell. I've run an autocontrol on this patient. That's just the patient's own cells versus the patient's own plasma, and it's negative, but I will talk about that a little bit more later on. And just at the bottom there, I have a positive control because this example I've done a manual panel, so you just put a positive control in just to make sure that it's worked. So we've got a 75-year-old male. They were last transfused 6 years ago and they've made an antibody. So I'm not quite done because I've thought, right, that's really looking like it's got anti-D there, but I need to have 2 positive examples of an antibody to confirm it and 2 negative examples of an antibody. So here you can see I've definitely got 2 positive examples and I've got 2 negative examples. So I can say that anti-D is here. So I've said the patient was transfused 6 years ago. So now I need to know what the phenotype is, because I need to know whether this is an allo or an auto antibody, because that's going to help you determine the clinical significance of your antibody. So now we're moving on to phenotyping, and I've included genotyping in this. I won't go into much detail about genotyping, but it's just to make you aware of what it is. So your phenotyping, this is performed using your unknown patient's red cells with known antisera. So this helps us to identify what antigens the patient has on their red cell. But if you do genotyping, it's actually performed using the patient's DNA, which is extracted from white cells. So phenotyping shows which antigens are expressed on the red cell, and this can be useful in paternal phenotyping too, if you want to know the predicted phenotype of a baby, for example, using the paternal sample. And a genotype shows which genes are present, but they may not necessarily be expressed on the red cells, because if something happens downstream, it might not be inserted into the red cell membrane, for example, but it gives you a good, useful sort of predictor. Phenotyping can't be performed when you've had a recent transfusion because you'll have dual populations of cells. So to me, recent transfusion is 3 months or less. But genotyping can be performed using a recent transfusion. The reason for this is because you're using the DNA that's extracted from the patient's white cells, if they've been transfused, the donor cells are leukodepleted, so hopefully negligible white cells in there, so you know that you're extracting DNA from the patient cells. With phenotyping, the IgG pos DAT can cause false positives when you use IgG antisera, and we will cover that shortly. But you can do genotyping if your patient has a positive DAT. Genotyping can also be used to determine genotypes for hemoglobinopathy patients, and this is done at IBGRL because it's a specialist service. And you can also use it for fetal DNA as well, so it's a really useful tool. So going back to phenotyping, so when we're looking at IgM anti-sera, you've got your patient's red blood cells there, you've got your anti-M, and shown at the bottom there, this is an IgM anti-sera. Probably confused it a bit if I use an anti-M. Done on your neutral cards, you don't need that bridging antibody, you don't even need to do incubation, depending on what your policy is, and it's done at room temperature. So if you perform your phenotyping, if it's antigen negative, the antibody hasn't bound to its corresponding antigen because it isn't there. This shows that your patient is negative for that antibody. So if they'd made an antibody and the phenotype was negative, it's an alloantibody because that's against self. It's something they are lacking. But if it was positive, you do get agglutination, and that would mean that the patient had that antigen on their red cell. And that's what it would look like in your cards, because like I said, I'm quite visual, so it's what's happening. So what does a positive result mean on paper, but what does it actually mean when I'm looking at the physical cards? And you would also you would also include a control here. So I've got a positive control and a negative control, and then my patient results. So the top patient is negative for the M antigen. So if they'd made an anti-M, it would be an alloantibody. And on the bottom, that just shows what a positive one would look like. So looking at the IgG anti-sera here, we've got similar picture. So you've got your patient red cells. This time, anti-WA is an IgG anti-sera, so you need that bridging antibody. So you're going to be incubating it at room temperature for 15 minutes. Sorry, not room temperature, body temperature for 15 minutes. And this is what is going on here. So if you're antigen negative, you've got the patient's red cells, the IgG anti-sera, so that's your anti-WA there, and your bridging antibody, but there's no agglutination because there's no binding, there's nothing for the anti-IgG to bind to, so we're all good, you get a negative reaction. But if it's positive, the anti-WA anti-sera will bind to the red cell, and then the bridging antibody will hold them all together, and then this shows that the patient's phenotype contains the WA antigen. So that's how we use it for that. And this is what it would look like in your card. And again, I've included a positive control. You notice with what you tend to find with your IgG antibodies as well, when you're performing anti-sera, you actually use a weak example for your positive control, which is why you're sort of getting a 3+, because we use heterozygous expression, because you want to be able to detect the weaker antibodies as well. If your patient's been recently transfused, and this is regardless of whether you're using IgG or IgM anti-sera, and you're using whichever technique it is that you're using, you can't do it because the patient's been recently transfused, so you'll probably end up with mixed field reaction phenotype, particularly if your patient's antigen negative and the donor's antigen positive. It could look like this, but you wouldn't actually know if it was the patient who was antigen positive or the donor, so you just can't say. So that's why we don't perform phenotyping on recently transfused patients. So going back to the patient we looked at there, they've detected anti-D. We don't know what the phenotype is at the moment. We're going to perform phenotyping, so I've just demonstrated how you would do that there. This helps to determine the clinical significance. We've said they can't be performed when they've had a recent transfusion, but our patient hasn't been transfused for 6 years, so there's definitely going to be no donor red cells kicking around. The autocontrol's negative, even though this isn't IgG anti-sera, it's just worth bearing it in mind. And in this case, we can actually use with ABO and D phenotyping for this patient, so you don't have to go and do a separate one. Sort of, here's one we did earlier because you've done it as part of your blood group. So you can see here the patient's D negative. So this means that the patient's red cells have not bound, there's no agglutination with the anti-sera, and it's negative because the antigen isn't there. So that it's an alloanti-D. So this is how we've realised that it's an antibody against non-self. So it's useful to know your clinical significance of your antibodies. So you've got different things here. I was a big fan of the antigen fact book when I was in the lab, would quite often be in there having a look. You've got various guidelines, and wherever you're from, you would have your sort of country's guidelines if you're not from the UK as well. And this is a clinically significant antibody can be defined as one that's capable of causing accelerated red cell destruction or even HDFN. This is why it's important to exclude them. So if you look at the BSH guidelines, anti-D is a clinically significant antibody. So once you've identified your antibody, it's worth checking your guidelines to see if it's clinically significant or not, because some antibodies aren't, so you don't need antigen negative blood. But in this case, we do. There's also information here of why you might perform an IAT cross-match. So when you have alloantibodies, your patient's not eligible for electronic issue. And this is just a recommendation to provide reassurance that the phenotype of the donor red cells is correct, because you might inadvertently pick out the wrong unit and then your IAT cross-match will be incompatible, which might prompt you to think, have I selected the right cells? It can help you to detect additional antibodies, or there might have been masks that you've not actually excluded in your antibody ID. And it just gives you reassurance that your antibody specificity is correct as well. And there is a note here saying if you found an RH antibody, you should really do an extended RH phenotype, provided it doesn't impede your provision of blood. So our patient's got an anti-D, it's clinically significant, it needs that RHK, the auto controls neg, and I've just mentioned there, just be mindful of transfusion history and include a control way to make sure your results are valid. So we've done an RH phenotype for our patient here. You can see the ones which are positive are big C and little e. So there's been direct agglutination there. This patient has those antigens on their red cell, but they don't have those antigens. So they don't have big C or big E or big K. So that's how you would see. And then we've got a phenotype there. Fab. So the next poll, please, Anna. What blood would we need to select for this patient? So are you just going to get random OD negs? Are you going to get OD neg, big C neg, big E neg? Are you going to give D neg, big C neg, big E neg, K neg? Or are you going to give them D pos? Or you might just not be sure. Are you all right to launch the poll there, Anas, please?
Yep, so I've launched the poll already. About 30 odd people have responded.
Oh, fab. Sorry, I can't see it. I don't know why it's not coming up. Just have to let you tell us what the... I don't need to have trouble loading my messages, so I do apologise about that.
So about 3/4 of the people have said group O, D neg, C neg, E neg, and K neg. About 11% have said group O, D neg, and following that, 8% have said they're not sure, and the other 8% have said group O, D neg, C neg, E neg.
Fab, thank you. So for this one, what you would be needing to select would be your D neg, big C neg, big E neg, K neg, because we are matching the patient as well. So this is in accordance with the BSH guidelines. So it's just saying, red cells should be D neg and K neg, because it's useful and it's easy enough to give K negative blood as well. And patients with anti-D who are little r phenotypes, that's your D, big C, big E neg, should receive little r blood. So you can see so far how we've used all those tools. So we've considered our patient history, the screen, the grading, your antibody ID. We've done inclusions and exclusions. We've used our panel cells. We've used Enzyme. You've hopefully seen how that works. We've looked at our panel sheets. We've looked at the clinical significance of antibodies, and we've even sort of looked at phenotyping and genotyping. So what I'm going to do now is just go through a few more grading examples with you. Then we're going to look at the direct antiglobulin test and an elution as well. So these are ones I've already done earlier. So this is an anti-M. So you can see there, and I've got some arrows on. So hopefully you can see, if you're looking at your results on your IAT column, you have stronger reactions where it's the double dose expression. So that's that homozygous expression, whereas you get a weaker reaction when it's paired up with its sort of partner. So they're called your antithetical antigen. So M and N, you can get those together. So you can see there, you do have your reactions. And sometimes it can even miss them, those heterozygous lines like I showed you before. And anti-M is sensitive to papain. So just when you're confirming the presence of an anti-M, it's good to know that it can actually it's react by IET. It's sensitive to papain, so it disappears in your enzymes. And it may even be that it's not hitting the heterozygous lines. So what I've got here is an example of another anti-M. So this time it's shown dosage and it's a cold antibody. So if you look at that very bottom line, we have a heterozygous line at the bottom, and it's actually missed it. So what you can do is anti-M's a really good cold antibody. So you can actually do a cold panel, and you can see there by cooling everything down that we've actually enhanced the reaction. So it's thinking about how those antibodies behave, which can just be really helpful. This can also, if you've got a cold antibody, one of the clues might be in your group. So it can actually interfere with the reverse grouping, because the reverse grouping cells are donor cells. These could be M positive. And in this case, it's interfering with your reverse group. So if you get sort of a few reactions where you think, this is looking a little bit like an anti-M, but it's missed a few lines, but I've got an interference with me grouping, then it could be just consider a cold antibody. Another example that we have here is your papain enhanced. So we've got a big C, and what you can get here, you can see it's papain enhanced. You haven't got you've got sort of enhanced reactions because you've removed some of those antigens, they're not getting in the way. But you can also, and you can see on line 4 there, you can even see dosage sometimes in your RH antibodies as well, but you tend to see it in IET and not so much in the enzyme, as you can see there. So I've just pointed that one out there. So the next one, we're just going to have a look at some multiple antibodies. So again, I've sort of already done these for you. In this one, I've gone a little bit backwards about it, because normally I would do my exclusions, then my inclusions, but I've done it the other way around here. So if we look at our big E, for example, we have different strengths of reactions when you look in your panel there. So I've sort of highlighted those. It's enhanced in enzyme. So you can see line 5, you've got a 2+ reaction by IET and a 4+ by enzyme. That's good because we know it's an RH antibody and it can be enhanced. Then we'll move on to the anti-M. So you can see again, this has missed the heterozygous line in the bottom there. So it's reacting as you would kind of expect a relatively weak anti-M to react. And you've also got an anti-big K there as well. And that generally the reactions tend to be about the same strength. What you may have noticed is some of the arrows are multicolored. So this one, for example, if we look at line 6, for this one, you have the cell is M and K positive, and sometimes if you have multiple antibodies on a line, using your grading, you might actually have stronger reactions. So that's where the grading comes in for that. So if we want to look at our exclusions for this now, I've sort of gone through and I've done the exclusions, and I'm looking and I can't exclude anti-D and I can't exclude anti-GKB. So I'll just leave this up here for you, but if you could launch the next poll, please, Anna. So I just want to know, can you exclude anti-D on this panel? So you either can, because it's not been masked by any other antibodies. You can, because you can actually exclude RH antibodies by enzyme technique, or you're not able to exclude it. And again, I can't see it, Anna, so if you could let me know just when it's available, thank you.
Sure, no worries. Okay, so from the 30 or so responses, now 40 responses that we've got, about half have said yes, RH antibodies can be excluded by enzyme technique. 40% have said no, you're unable to exclude anti-D on this panel, and a few people have said anti-D is not being masked by any other antibodies.
So thanks, Anna. So for this one, your anti-D is being masked but in IET by the other antibodies. So you've got your M, your K, and your big E. But if you look at that line 1 in your enzyme, that is actually negative. So those of you who have said RH antibodies can actually be excluded by enzyme technique are correct. But these are things that nobody expects you to know this off by heart. These are things when you're learning how to do things, you would use your guidelines, talk to your colleagues and things like that. So in this case, you can actually exclude your Rh antibodies by enzyme, and this is lifted from the BSH guidelines. It's acceptable to exclude Rh antibodies using a validated techniques incorporating enzyme-treated cells. So you can in that case. Now the next poll, can you exclude anti-JKB? So you either can, because the cells which are negative for the other antigens are homozygous, or you can't because the cells which are negative for the other antigens are heterozygous. So if you can just let me know when that one's launched. Thanks, Anna.
Sure, I've just launched the poll now. Okay, so most people at the moment, we've only had a few responses, are saying no, you cannot.
Yep, and that is the right answer, Fab. So no, because the cells are heterozygous. So if you remember earlier on, we're saying you can only exclude KIT antibodies using heterozygous expression. And just to highlight that here for you, the two cells, so lines 4 and 8, are actually heterozygous expression for JKB, so it's single expression, so you wouldn't be able to use that cell to exclude. So we can exclude anti-D, we can't exclude anti-JKB. Would need to test the big E and K negative cells against homozygous JKB to be able to exclude anti-JKB. For the other antibodies that it's looking like we've detected, we need at least 2 positive and 2 negative examples to confirm their presence. Or if the patient had already been seen and we had a phenotype on them, you can just confirm it with 1. But depending on your transfusion history as well, we're saying we need to know the phenotype of the patient. So you either have it already or you don't have it and you're going to have to perform it if the patient hasn't been transfused. So this is why we need to know the patient history. So for this patient, we've got a 60-year-old female. They've come in for a hysterectomy. They've had multiple pregnancies. They're grouped as OPOS. They've got no record of historical antibodies. The patient has never been transfused, so probably just made these antibodies during pregnancy. And 2 units of red cells have been requested. So when we're looking at our inclusions now, we need to see if there's 2 positive examples and 2 negative examples of each. So in terms of your K, we've got 2 positive examples which are negative for any of the other antibodies that are there, and we'll have 2 negative examples. You wouldn't be able to use cell 6 because that's also positive for anti-M, so you couldn't use that cell to confirm. Then when we're looking at the big E, you have, I feel it's missed something out here, 2 seconds. There we go, we've missed an M, I knew it was missing. So we've looked at the K, now we'll look at M, you have 2 positive and 2 negative examples, but you couldn't use cell 3 and you couldn't use cell 6. Skip past that one. And then looking at the big E, you only have 1 positive example. You've got 2 negatives that are, again, negative for all of the other antigens as well, but line 3 is positive for M, so you couldn't use that cell to confirm the presence of big E. So what you can do is put some more panel cells up, or you could look at your screening cells. In this case, we just happen to have selected cells. They're negative for all of the other antibodies. So we've gone wild here. We're also using this as an opportunity to get rid of anti-JKB. So you can see cell 1 is negative for big E, M and K, and it's homozygous JKB expression. We've got a negative result, so I can exclude this. But because I like to do it twice, I've actually put another cell up there. So that's JKB gone. We've got rid of that now. And I can also use cells 3 and 5 because they're negative for M and K to confirm the presence of my anti-big E. So it's using all those tools and using the guidelines and things to work out what you've got. So what we have now is we've definitely got anti-big E, which is clinically significant. We've got a big K that's significant, and we've got an anti-M, which is coming through by IAT. So we need to give antigen negative blood. As with the anti-D, because they've made an Rh antibody already, it's a good job to do the phenotype as well, just so you can prevent them from producing any more further Rh antibodies. But what we don't know is the phenotype of this patient. So we've said we've never seen them before, so we've not done the phenotyping. You can't use the grouping this time because it's not the D that we're necessarily interested in. It's the other antibodies. So we are actually going to have to do some phenotyping. So we know that this patient's never been transfused. Again, I've put a note there that the autocontrol's negative, just as a sort of prompt to yourselves, just to be mindful of it. The patient needs big E, M and K phenotypes and in addition an extended RH and K. The autocontrol's negative. So this is the patient results here. So we've got agglutination in our big C, D and little e and our positive control. There's no agglutination in the little c, big E and our control. So this patient, these are alloantibodies because the patient's lacking the antigen on their red cell, and we've already seen that they're clinically significant. So what blood would you select for this patient? If you can launch the poll, please, Anas.
Okay, so the poll has been launched.
So really we've got here, we've got the D pos, your big E, K, M, so matching for the antibodies, or you're going to give your extended RH phenotype, or you're going to give D negative, big C negative red cells for this person. How we're looking there, Anas?
So about half the people have said option 2, so that's group O, D pos, little c neg, big E neg, K neg and M neg. About 30% of people have said group O, D neg, big C neg, big E neg, K neg and M neg. And a few people are not sure, and a few people have said the first option.
Yeah. So for the first option, you could give that, but ideally you'd want to be given the second option because you're fully matching for the extended RH because they have an RH antibody. If you gave option 3, which is the big C negative, chances are that unit is going to be little c positive. So you may want to avoid that one because you wouldn't want to stimulate an anti-little c. So that is based on the guidelines, that is the blood that I would select for this patient. So thank you for taking part in the polls there. I'm just going to finally whiz through, and I've only got 10 minutes left and I've still got a lot of slides to cover, but we're good to go, don't worry. So this one here is just a final example of a papain sensitive. You might see there on line 7, we've got a W instead of a positive. And what you might see on this panel, we need to remember these are actually made from donor red cells, so the patients. So this happens to just have a weak expression of the Duffy antigen on there, just to let you know. And in this case, you can see there it's papain sensitive. So you have your negative reactions in enzyme, so that helps to confirm that you've got that Duffy A there. But in this case, we've actually got a positive autocontrol. So when we're testing the patient's autocontrol, this is the patient's red cells versus their plasma. The positive autocontrol in a patient who hasn't been transfused, it can be due to autoimmune conditions, might be some medication they're on, or it's just one of those things. Sometimes you do just get like a DAT pos patient, it's 1 in about every 8,000, I think. Just one of those things. In a positive autocontrol in a patient who has been transfused might mean that they've been sensitized and they've produced an antibody, and that's actually antibody that's bound to those red cells on the donor red cells, and these can potentially cause transfusion reaction. So it's important to identify antibodies using that information from your DAT because they might not be present in the plasma. So we'd need to perform a DAT, and if it's IgG positive, we're going to look at doing an elution. And I've just got a note there, sorry, that I've skipped past, just to say, if your patient has hemolysis or clinical signs of a transfusion reaction, even if the DAT's negative, you would want to be doing an elution on that. So what is your direct antiglobulin test? So this is what's showing you what's bound to the patient's red cells, so in vivo. It shows you if there's IgG bound, if it's IgG positive. If your patient's been recently transfused, this is a mix of those patient and donor red cells. And if they've been transfused in the last 6 weeks and IgG pos, we'll need to be doing an elution on that. C3D positive shows that there's complement bound to your red cells as well. And whenever you're doing a DAT, you need to consider your patient history, clinical data, and results of your other tests when you're interpreting them. So when you're doing a DAT, essentially what it is, you've got your patient red blood cells which are sensitized with either IgG and or C3D. if the patient's been transfused, this is not only patient cells, this is donor cells that are going to be sensitized with these as well. You add your AHG, don't need to, excuse me, incubate, you can just centrifuge these. And you can see there the bridging antibody is going to cause agglutination, which is why you get a positive DET. Excuse me. And this is what it looks like in the card. So you can have just IgG, you can have just C3d, or you can have both, and your control should be negative. So going back to the phenotyping, so you'll remember that I said you can't do IgG phenotyping if your patient has a positive DET, and this is why. So you've got your patient red cells sensitized with IgG. When you add your Duffy antisera, the antigen isn't there, so the anti-Duffy will not bind. However, the anti-AHG will bind the IgG on your patient's red cells and cause agglutination. So you actually get a false positive result. So that's how this works. So there's no Duffy antigen there, but you get a false positive because it's binding to the IgG on the cell. But if you'd use an IgM antisera, you don't need the bridging antibody. So there's nothing for the bridging antibody to bind to. If you are, not that you're doing it, but it can't bind the IgG. So you don't get a false positive, you just get a genuine true result. So this is if the patient hasn't been transfused. What about those patients who have been transfused? So this is when we're getting into elution territory. So to do an elution, the IgG pos and recently transfused, this has shown that antibodies are bound to the patient's red cell, but remember it's a mix of donor and patient red cells. So if your patient hasn't been transfused, there's no donor cells to elute from. That's why we ask for the patient transfusion history. You perform an acid elution technique, and what this does is, if you think of the antibody is so new, it's bound to the patient's red cells. It's not necessarily free in the plasma, so you need to unlock it. So we use a process called acid elution, and it just sort of removes them from the red cells and brings them into a solution that we can test. So I'm going to show you how we do this now. So this is a photo, several photos of me actually doing an elution in the lab. So you take a ml of your patient's red cells. So this is going to be, remember, it's a patient sample, but it's going to contain donor red cells. So you're taking those a ml of packed red blood cells, you give them a really good wash. And what I mean by washing them is you add saline, centrifuge it, take off the saline, put fresh saline in, give it a mix, centrifuge it again. So you're washing any residual plasma because you want to see what's bound to the red cells, not what's in your plasma. So once you've done that a few times, you produce this sort of supernatant. You keep a little bit of the supernatant because you want to test it to make sure that you have washed the cells properly. But now what we do is you get this acidic solution, and this is just the kit we used in the lab when I was there. And you add it drop by drop. So if you had 10 drops of patient's red cells, you'd add 10 drops of the acidic solution. Then you centrifuge it. Then what you get is, this time you want to keep that supernatant. So you're keeping that acidic supernatant, but you need to put a buffer in it. So what we do is there's a buffer and it's blue, and you add it again drop by drop. And once it reaches the point of neutralization, it turns blue, and you've got to neutralize it because it's acidic. And if you didn't neutralize it and you put it in your red cells, it would lyse them. So your result would be invalid. And then you just treat it as though you would plasma, and you do your antibody ID. So going back to that first example, we've got patients, they were transfused, they hadn't been transfused, sorry. They made an antibody ID. They didn't have made an antibody ID, they'd made anti-D. Sorry, I've getting a bit confused. They've now been transfused and they've sent another sample to the lab. So this is the results. So same as the last time, we've got that anti-D there. The difference this time being is that the autocontrol is positive and the patient has been transfused. So that we've done a DAT that meets the criteria for it's IgG, so it meets the criteria for elution. So we've done that elution process, and I've made this blue. And you can see there they've actually got an anti-GK, but you notice this wasn't in the plasma. So this is showing that there's a brand new anti-GK stuck to your patient's red cells. So that is what happens when we do an elution. And that last column there, I've got, it's LSW, and that just stands for last washed saline. So we test that as well, just to make sure that I've actually, when I've performed the process, I've actually washed it properly. When I'm thinking about the blood that I want for this patient, they've made an anti-D and they've got an anti-GK, which is clinically significant. So thinking about this, same criteria as before, they've got anti-D, but want little r, and you want to give them big K neg. if the patient's big K and neg. But for the anti-JK, they've been transfused, so we can't do the phenotyping this time. But it needs antigen negative blood because it's clinically significant. The antibody would be reported as not specified and would put a note on to say do a phenotype 3 months post-transfusion. So hopefully with 3 minutes to spare, we've covered everything today, but you can see how everything just interlinks. So it's all those little tools on their own are great, but together they just work really well to help you with your antibody identification. We're doing another session in, we'll send the dates out soon. So what we're going to do is we'll do a quick recap of what I've covered today, but I'm going to do some case studies. So if you've got anything, if you want to quickly pop anything in the chat, if there's anything you want to cover over the next 2 sessions, either drop us an email at the OnlyCells address or or pop it in the chat, and we can use these as an opportunity for you to cover some cases. So it's just putting into practice what we've done. And then the final session that we're going to do, we'll be looking at absorptions, high incidence antibodies, all the kind of thing that you might not necessarily see as a routine investigation in your laboratory. So hopefully that'll be helpful. And I've got 2 minutes left. I was going to ask what your favourite antibodies were, but we don't have time. If there are any questions, if anyone has anything. And sorry, because I always take too long on these talks, but it's a lot to do in an hour. Thanks.
So what we can do is if people post their questions in the Q&A section at the top or in the chat, then we can address them later. So I'll extract them and we'll provide them with a certificate at the end. Because we've only got a couple of minutes left, Helen, thank you very much for such an informative and engaging session. I've got to admit, though, I never thought SpongeBob and Antibody ID would end up in the same slide, let alone in the same context. But that's why I really enjoy your talks. You always make you always make the complex topics relatable, memorable, and fun to learn. A big thank you to everyone else who joined us today. Throughout the meeting, I received a lot of emails and messages from people saying they weren't able to join at the beginning. Apologies for that. I'm not sure what happened with Teams. So for anyone who missed the beginning, this will be uploaded to YouTube. So you'll be able to watch it at a later time, which is convenient to you. I'll be sending out the certificates of attendance over the next couple of days, along with details for the next session in the series. And if you attended as part of a group, then please drop me an email with details of individual attendees so I can make sure everyone receives their certificate. And if you haven't already done so, I'd encourage you to register for the OnlyCells webinar mailing list. And that way, whenever a webinar is organized, you'll get a detail sent directly to your inbox. And if you're on your desktop, you can click on the mailing list on the slide and it should open up the link, or you can scan the QR code. So thanks again for joining us today, and we look forward to seeing you on the next one.
Thanks, everyone. Bye.